Yeah, hello everyone. So, any doubt in the introduction session?
No doubt.
Okay,
No, ma'am.
okay.
No doubt.
So, in today's session, we are going to see how to download the raw data from the NCBI SRA database.
My Linux is not working. I did
No,
not. I
no,
was not able to install WSL.
no problem, sir. I will guide you after the session. Okay, because there are maximum people. We cannot
Okay.
interrupt the session for them. Okay, we have to run very smoothly for those people also. Okay. So
Perfect.
maybe at like yesterday also I told you you can ping me anytime before the session. You
The problem is
haven't
that I
texted
work.
me.
I
Okay,
work.
no problem. Whenever you are free, just ping me. Okay, I will fix that problem. Okay, okay. So this is one of the database from where we are going to download the dot data. So you have to put any keyword search here. So for example, we are working on this RNA-seq. So I will just write here RNA and then search. So I will clear all the filter first. Yeah. So you can see this many hits you are getting total, right? So on this side, if you want to get the raw data from any specific organism, you can select either human, mouse, Drosophila. It's up to you. Okay. According to your data, you can just select any specific organism from this. Then on this left hand side, there are some filters again. And in the assess, there are two options, control and the public. So control assess means whoever has submit the data, they don't want to share freely. So we have to ask the permission from the owner to download the data. Okay. It is not of free of cost here. But in the public means it is freely available and you don't have to ask any permission from anyone. Then source, either DNA, RNA, it's again up to you. Then type is exome. If you want to work on entire genome, you can select the exome. Then library layout. We have discussed in the introduction also, there are two type of sequencing we are doing, the paired end and the single end. What if I'm interested with the single end? So I will just select the single end. And as this application is RNA based, so I'm going to select this RNA. Now the platform. So platform is nothing. These are some sequencing machine. Okay. According to your sequencing platform, you can download the data and you can just scroll whatever the data it is belonging to the Illumina, you can just search it. Again, the strategy and the data are also stored in the cloud also. And the type of files you will get in the form of FASTQ also and the BAM also. So these are some alignment information you will get here. And if you want to start from the reads, you can just download the FASTQ files. So whenever you are downloading the data from the NCBI SRA, so just make sure you never go with this updated, okay, this first page. Why? Because it is having the incomplete information, still the research is going on for this particular data. Okay, whenever you try to download this data, you won't get the complete information. So try to go with the older data.
Okay.
So for example, I am going to choose this. So you can see this is the SRX ID. This is just a set of lab experiment they have performed. You can see the Homo sapien they have taken and they have performed the RNA-seq on this data. So which platform they have used? Illumina. So one time the Illumina machine was used, how many spots have been used in the sequencing machine? That spots they have listed, the basis and the total size of your data, that GB download you can see here. Then it is submitted by the NCBI. Then study, what type of study they have done. If you want to,
What is sport here?
which one, ma'am? What, ma'am?
Ma'am, sports. What is sports meaning here?
This, ma'am. In the Illuminats, there are some spots present there. So how many spots? This is 4.3 million they have used there. Okay, spots.
Ma'am, is it similar to flow cells or like is it
Yes,
different
ma'am.
than
Yes,
that?
sir. Flow cell only.
Okay.
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 So if you want to study more about this data, you can just scroll down to the abstract, then the sample they have listed, and the library. This is the RNA-seq single length data. So how to download this data? Don't try to download this. Okay, I've already shared the data in your system, like in your WhatsApp group also in the drive. So I'm just telling you how to download that. So for the single length data, you can see this is the FASTA and the FASTQ download here. So you can download the FASTQ file, right, for the single length. So it's very easy to download the single length data from the NCBI SRA. But what about the paired end? The question is that. Again, I will just clear all this filter. Then I will select this paired end data now. Now again, go back to the last, the older data. Then open any one of the entry. Then you can see it is belonging to the mouse. Okay. Then this is the paired end data. Now, if I want to download this data, just go to the run entry here. Go and select the FASTQ. So can you see, like in the introduction also, I told you in the paired end, you are getting what? The two files. So in the paired end, what happened? The adapters are ligated at the both the ends, and the sequencing machine, what it do? It is sequencing from this side also and this side, right? So here you are getting what? The two files, ID with underscore one, which is representing what? The forward file. Then other file is your reverse file, right? So two type of files you will get in the paired end. But here in the SRE database, you can see when you download this FASTQ file, you are not getting the separate files, right? For the forward and the reverse. So it's very difficult to separate this forward and the reverse file. Okay. So how to download this paired end data? What you do, just simply copy this SRA ID here and go to one of this database, that is the ENA. Then search this ID. Again, same information you will get here also in the ENA. This is the paired end data, and you can see Illumina and the mouse here. Okay, if you scroll down, you can see the two files here. So this SRA underscore one is what? Your forward file, and this is your reverse file. Are you getting this?
Yeah.
Did you
Yes,
got
ma'am.
how to download this paired end data now?
Yes, ma'am.
So it's up
Very
to
helpful.
you guys, like whether you want, you can download the data for the single line from the ENA also. You just have to simply put the SRA ID here. You will get that. Okay. Now I've shared this data. So everyone is having this data, RNSec data, this zip file.
Yes, ma'am.
Okay. So
Also, it's in the WhatsApp group.
yeah, sir.
Okay.
So from where this data has been taken, I will guide you for this also. So I've shared the link in the chat box also. And the data which I've shared on the WhatsApp group or maybe in the drive, the data has been taken from this link. So you can see again, they did what? The differential gene expression here. Illumina platform they have used, design submitted by this department. And if you want to read more about this study, you can study at your home this abstract part. Now, this is the,
Professor, sorry,
yeah.
is it possible to reshare the file because I have no access that and I cannot see the file
Oh,
in the WhatsApp group.
okay, just wait, sir.
Okay, thank you.
Yeah. Do you have the access for the drive, guys?
Yes.
Everyone?
Yes, ma'am.
No, ma'am.
Who's this?
No, ma'am.
Okay, just wait. Okay, so this data you can see it is in the compressed format. So what you have to do, you need to extract this. Hamid sir, like the drive link has been pasted there in the group. You can see your WhatsApp group.
Yeah. Now I'm starting to download it.
Okay,
Thank
so
you.
everyone download this raw data and then you have to unzip it.
You are going so fast so that we did not see. Anyway, I did not see how you extract.
Okay, just wait, sir. Do you have 7-zip, sir, in your system?
No, I don't think.
Okay, so what you have to do, see, just select this, then
Yes.
right click it, then show more option, then
Yeah.
7-zip.
Okay.
Then you need to extract here.
No, I think I should install 7-zip first.
Okay, so you can install the 7-zip. So meanwhile, you can see in this raw data, we are having 4 files: control 1, control 2, treated 1 and treated 2. Okay. So if you open this RNA script, so the link I have pasted, okay, in that script from where the data has been taken. And I want to tell you this control one, I've taken this ID. So how to search that? You can see here. Go to all runs.
Sorry, what is the like difference between the all experiments and all runs here?
Where, sir?
What is the difference between all experiments and all runs in the portal that you just opened?
This one?
Yeah. So like
Here.
the all experiments and all runs. Yeah.
No. It's in the all the runs. Actually, it is including the sample only, sir. Okay.
Okay.
So you
Okay.
can see here, if like I have opened this all run from here, so it is not showing any sample. Can you see below,
Right.
right? So what I did, like instead of using this, just select any one of these two. Okay. You will get the sample below. Yeah. Can you see
Yes.
below now?
Yeah. So multiple samples. Okay. Got it.
Right. So you can see here in the script, control one. Okay, I've taken this 458. So you can just search it 458. Okay. So if you scroll towards this is the control sample. Can you see here? So this sample I've taken as the control one. Then in the pipeline again, 459, I've taken the as the control two. This one. Okay. Next, what is my treated one? You can see 452 ID. This is my treated one and this is my treated two. Are you getting this?
Yeah.
Okay.
And
So next step, how to download this data. Okay. So if copy this ID, go to the ENA browser here. Now again, same information you will get here in the ENA also. And you can see the link, okay, for this treated one. So if you want to run or download this file from the terminal, okay, why terminal? Any idea? Sometime you are downloading from this also, just putting and just selecting from here, right? So there may be a chances like your system gets stuck in between, right? And the download is incomplete. So in the end, just like we always prefer the terminal based. Okay, so what we have to do, just copy the link address from here, and then how I've created this, you just have to type wget -c and paste that link. Okay, then you need to copy this and you have to open the terminal WSL.
Professor, sorry, it was too fast and I didn't understand well.
Okay, just wait,
Yeah.
sir. So I will create new folder. So what I did, so this is my treated one sample, sir. Okay, what I did, I just pasted the ID here in the ENA. And if you want to download this, what you have to do, just copy the link address. Okay, then I want to download the file treated one in this new folder. Okay. So make sure you open the WSL from the new folder, and I will type here the command wget -c, and I'm going to paste that link. Okay. Then it will download this file. Can you see? Did you got this?
Yeah, I got. Just I want to try that.
Okay.
It's
So at
difficult to try the same time we are working on it. Oh my God.
no. So what you do after the session, you can try if you want and do, like, because this is only for 1 hour session. We cannot stretch for 15 days, right? So you will
Yeah.
get the all the recordings. But if you have like any problem, you can share me tomorrow also, right? Anything if you want to ask.
Can I ask a question?
Yeah, sure.
What's the difference between curl and widget?
Which one?
When I saw you type widget and you paste the link that
You
have
can do with the curl also, sir.
we curl. Okay, thank you.
Yeah. Okay.
Hello.
All the commands. Yeah, ma'am.
Ma'am, so I have to do all this in Anaconda?
No.
No?
Don't
Okay.
perform these commands, guys, because I've already
Okay.
shared the zip file with you. So if you
Okay.
see here in this, the data is very huge. Can you see the size of this?
Yes.
Right. So what I did, if you try to run this pipeline, okay, if you take this original data in your system, it is going to take lot of time. It might happen your system gets stuck because sometimes configuration is not supporting. You need to have the server, okay, where you can run the huge file. So what I did for your data, I have cut down the data.
Okay.
Can you see? I have used this command head. Then these many reads have been cut from this control 1 file, and then this is my file you are getting in the raw data. Okay, control 1. Can you see this in the raw
Yes.
data? This one.
Yes. Ma'am, can you please tell me when which of these steps I have to do in Anaconda and not in the other app?
Just wait for the Anaconda, and for
Okay.
the window user also, you have to perform from these steps. You guys are
Okay.
already having this file. I have already shared this control 1, treated 1, treated 2. I've already shared this in this
Okay.
raw data. Can you see that?
Yes.
You just download this file first, raw data
Okay.
in
Yeah.
system. I'm just telling you from where this data has been taken. Okay.
Okay.
Treated one and treated two, or control 1, control 2 are two independent experiments, or they are like parrot sequence, pair and sequence. I don't know.
Sir, this is your data. I've shared this link also with you. Go
Okay, thank you.
just
Yeah,
wait.
it's fine. It's fine.
Just check it from here also. This is your single end data.
Okay.
Okay. That is why you can see here in the script also, you are taking what the 2 replicates for your control and the 2 replicates for your treated. Okay. So in the introduction also I told you we need to take the replicates. Okay. So that is why I've taken this control 1, control 2 and treated 1, treated 2. Are you getting this? How to
Yeah.
download the data? Okay. So whatever the data I've shared with you, I have cut down the reads. Okay. These many reads have been cut down, and I have assigned this particular file as the control 1 and this 459 as the control 2.fastq. this as your treated 1 and treated 2. Then after cut down the data, I've shared this zip file with you guys. This one. Okay?
Okay, Professor, you mean now we don't need to run any
No,
active
no,
code?
no, sir.
Okay, because
No,
you prepared. Okay,
yeah.
okay.
Already
Thank
I
you.
have prepared for you guys because it will take lot of time,
Yeah,
right?
exactly,
Okay?
exactly.
Now, so everyone is having this raw data now?
Yeah.
Okay.
Yeah, yeah.
So you can see after downloading this raw data, next step is to check what the quality, okay, of your raw data. So how to check the quality? So there is one tool.
First Q.
Yes, sir. Fast QC, right? So how to download this? First, I will tell you. Step one is to download the raw data. So which I've already shared with you. Okay. Then step two, we are going to perform what? The quality check by FastQC tool. So how to install this? Sudo apt-get install FastQC.
Wow.
Then, so this is like I'm going to install with the help of repository method. Okay. Then if I need to check like whether this tool is installed like perfectly in my system or not, I need to open the manual, that is fastqc-help. So in my system already the fastqc installed. So what you guys can do, any one of you like who is having WSL can share their screen so that I can guide you and other can also see.
I have WSL.
Yeah, Hamid sir, you can share your screen.
Okay. Okay, is it visible?
Um,
Ma'am, is it the same procedure for the Anaconda also?
just wait. For the Mac, I will tell you. Just wait, ma'am.
Okay.
Okay.
Just make sure Anaconda is installed. Okay.
Yes.
Okay, Professor, this is
Perfect.
the
Perfect, sir.
raw
Perfect. So what
data.
you do? Yeah, yeah, yeah. Just open this raw data.
Open, okay,
This
and
raw
in
data.
here,
Right. From
in,
here, just open the WSL. Yeah.
okay,
Okay. So we are going to install what the fast QC,
okay,
the repository method. So first we
okay,
need to check whether this tool it is present in the repository or not. Okay.
okay,
So just
okay.
type here sudo
Mm.
sudo space aptitude apti
AP,
ti
T, I didn't get AP,
t t.
D,
No. I will just send this command in the chat. Just wait.
okay, sir.
Yeah.
Then enter.
No.
No, it's
No.
it's
No. No.
it's yeah, it's error. Yeah, yeah.
Okay. Yeah. 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 The
Now
pass.
it needs password.
You no. Just wait. You need to put the passwords are there.
Mm,
Le père de
where?
Zino.
What is the password, Hamid sir?
I think 1, 2, 3, 4.
Okay, just put there 1, 2, 3, 4. Then enter. Okay, can you see everyone like the sudo aptitude command not found? Can you see?
Yeah, yeah, not found.
So this is the error here, right? Can you see aptitude? So what I told you, in the previous session, the aptitude is your package manager, which is storing all the information for your tools, right? So
Okay.
that means first you need to install what? The aptitude. Then only you can install the tools, right? So how to
Okay.
install that? I will just share again the command.
Okay.
Make it yes. Why? Enter. Okay. Now what you do again, just type
Right.
sudo apt, give the space apt.
Yeah, this that you.
Yes, yes, yes.
Okay.
Enter.
Space, space.
Yeah.
Yeah.
Now can you see after installation of your aptitude, now you can see the fast QC is there, right, in the repository method. So
Yeah. Yeah.
now we are going to install this fast QC. So what is the command, sir? Can you tell me? What can be the command?
Delegate?
No, 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 no.
No, no, no. Where can I see command?
So there are only two commands. So this is your first command for searching any off tool, sudo
Mhm.
aptitude search fast QC. So next command is what? Sudo apt hyphen get install fast QC. Yeah.
You
So
mean,
this is the two command for the repository. Okay. Remember that. So by these two commands, you can install any tool. Okay. Just make sure it should be present here in this aptitude. Yeah. Now it is installed. We need to see whether this fast QC is installed or not. So you just have to type fast QC space hyphen help
fast QC and
space hyphen
space and
help.
help
Yeah. Yeah.
justice.
Yes, sir. Enter. No, no, no. Hyphen.
Oh, sorry.
I've
Fast,
shared the command also.
fast QC
Space.
space. That you mean like this?
Not this, sir. Not this.
Mm,
Hyphen.
I can this.
Just copy that command from the chat, sir.
Oh yeah,
Yeah.
yeah. It's much better. Thank you.
Yes.
Okay.
Enter. Now, can you see the fastqc is installed, and after putting this command fastqc hyphen help, the manual is opened. That means this tool is installed perfectly in your system. Okay.
Okay.
Did you got this fastqc
Yeah, yeah,
installation?
yeah.
Okay. So the one who is having the Mac, so they can share their screen as well.
Okay. Should I stop sharing? Yeah.
Yes, yes, sir. Yes.
Okay. Thank you. Thank you.
Who was it? Surbi ma'am?
Yes, yes, ma'am, I don't have that raw data
Okay, yeah, Anil.
downloaded. So,
Okay, okay. Okay, no problem. He's also having the Mac, you can see.
okay,
Okay.
thank you.
So, did you downloaded that Anaconda script like which I have shared? Mm, okay. So, what you do, sir? Can you just browse it? Just, yeah. Now you have to type install fastqc by Conda. Just type that. Yeah, open the first link, scroll down. So here you can see the command here, Conda install bioconda this, right? Copy that and then paste. Yeah, enter. So Survi ma'am, this is the command, okay?
Yes.
Then after that, what you have to do, just type fastqc space hyphen help, okay? So that tool will be installed, okay?
Okay.
Yeah. So meanwhile, Anirudh, you can run this, and if you face any problem, just let me know. Okay then. What about others? Did you install the fastqc tool? Okay. So now, how to run this? You can see, like in this manual, there are some commands. Can you see fastqc? Then give your file name. So this is the command to run the fastqc. So just call the tool fastqc and give your file name. That is control1.fastq, control2.fastq, then treated1.fastq, then treated2.fastq. Or what you can do, second command is either you use this fastqc, put the star and dot fastq. So here in this command, we are calling the fastqc tool. So why I have written star? Star here, it stands for all. So right now we are only having the 4 files, right? Only 4 files. But what if you guys are having the multiple files? Okay, like thousand of files you are having with the dot fastq. You can see here in this also the dot fastq is common. Can you see? Right? So what you can do, it will just take all the files which is having the dot fastq extension. So it is going to run for all the files. Okay. Either you just type your file names, either you just put the common extension there. It is going to run the fastqc. So just make sure you can see here, this is my small letter treated, but in your file, you can see it is written in the capital letter, right? So it is not going to take the treated file. So make sure like this, Linux is very sensitive, so it should have the same file name as well. So it should be capital T here. Okay. Now what you do, just simply copy any one of these command, and from here you open the WSL where your files are, and then run this. Now see, it has been checking the quality of the treated one. So did you got this command? Any doubt till here? Any one of you?
I ran and it's running like your system.
Yes, sir. Yes, sir. It will run like this only. So whatever the files that is present here in the raw data, so it will check all the quality check for those fastq files. Okay, so it is see treated to here now.
Can I share my screen? There's just something that like I was also running and
Yes, yes, sir. Yeah.
uh, share now. Can you see my screen?
Mm, yeah.
So when I ran this command, in the beginning it just shows me null, 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 and started analysis and then null, null, null. So uh, this is not similar to what you were seeing, right?
Okay, just scroll down. Put ls here, sir. Okay. No worries, sir. This is not an error, sir. Okay.
All right. Thank you.
Yeah, it is perfect.
And Professor, this command is used when we have many sample, yeah?
Yes, sir.
Mm,
Like it's
yeah.
up to you, sir, whether you want to write your sample names. Okay, whether you want to run with the star, it's up to you. Why I've taken this, so that it will take like automatically all the FASTQ file, right? But if you
Okay,
want
yeah.
to put the all like the file name, it's up to you, sir. Okay.
Okay.
Okay. Now, yeah, you can see it is like has created the two files. Can you see for the control one, it has created the HTML file and the zip file.
Yes, exactly.
So this zip file is what? It is containing the summary of the graphical representation. So this HTML file is very important for the analysis of the FastQC. So let's open this control1.fastq. Yeah. Can you see this report, FastQC report? Now on the left hand side, so there are certain parameter they have checked for this data. You can see basic statistics per base sequence quality, tile sequence per sequence quality score. Now out of all these parameter, there are three important parameter is per base sequence quality, over represented sequence, and the adapter content. So you can note somewhere these three is very important: adapter, over represented, and the per base sequence quality. Along with that, you can see it is written like it is giving you here green take, red color, and the yellow. So green color is indicating the good quality, red is your poor quality, and this exclamatory sign, it indicate what? The average quality of your data. Now let's understand this basic statistics. You can see here the file name, it is giving you control 1. So for which the raw data you are checking the quality, that file name they have listed here. Then next is the file type, that is what the conventional base call. So actual base calling method they have used for each and every base here. Okay. Then encoding, they have been performed by Sanger and the Illumina. So encoding means what? The conversion of the Fred quality score to the ASCII character. So they have used these two sequencing machine. Then total sequence here. Number of reads in our sample is this much in the control 1 file. Then sequence flagged as poor quality, it is 0 here. So out of total reads, no read is having the poor quality. So that is why here written 0. Then sequence length. So most of the reads in the control ones, it is having what? This much length. Okay, 75 around 150. Then GC content is also good. It is 51 here. So it should be like between the 40 to 50. Okay. Then next, did you got this basic statistic?
Yeah.
So for the encoding, does that mean that, like, I understand that this is just one sample and it has been performed on Illumina,
Yeah.
but then they have stated it as slash Sanger. So does
Yeah.
that mean like the fret quality or like the coding that is used in Sanger as well as Illumina is same?
Just wait. So this FastQ file which you are getting, okay, the one you have downloaded, they are containing, sorry, 4 lines equal to one read. So what are those 4 lines? First line start with the ID. In the second line, you will get the sequence. In the third line, you are getting a separator. Just wait, it is. It's a separator, sir. So every platform it is having different separator. For example, for the Illumina, there is a plus sign. So what it will do, it will separate the second line and the fourth line. Why? Because in the fourth line, there are some sky characters which is listed like this. Okay. So you will get something like this, sir, a sky character. So why they are converting? Any idea? Any idea?
Not really.
Actually, this
Oh,
sky character, whatever you are getting here in the fourth line, it is representing your Fred quality score. Okay. So when you have a number, it will create a huge file size. Okay. You already know like the one read, it is containing what? The 4 line, right? So that
right.
means it is a huge file, that FastQ file. So there is one method that is the encoding. So encoding, what it do, it is just converting the score, okay, or maybe the Fred quality score that we have discussed during the important terms also, into this sky character, sir.
Got it.
Got it?
Yes,
What are those?
the fret quality.
Okay. Now, this is very important per base sequence quality. You can see here on the like x-axis, this is your one read here. Can you see the length of the read? It is having what? The 150. So this is one read here. Okay. And on the y-axis, it is indicating your Fred quality score. So you can see here, they have like this graph has been created into 3 different colors: green, yellow, and red. So this green color, it is indicating what? If your bases are falling here in the green, that means the quality of your data is good. If it is orange, then it is average quality. But if the base is falling here in this red, that means it is having the poor quality. And also for this particular read, can you tell me what is the Fred quality score here by looking at this graph?
Ma'am, below 20.
Below 20,
Ma'am,
Sakshi?
36.
It's
ma'am,
it's
for the red
30,
one, it is below 20.
it's more than 30.
it's
And ma'am,
36.
for green one, it is above 30.
Who is 36? Who is this?
Anirudh.
Yes, sir. Correct. It is 36. Can you see that? The line here, it is falling in the green color. Okay. So that means on the y-axis, it is indicating the Fred quality score, and you can see it is more than 36. Can you see? So that means for this particular data, the quality is good, and also the Fred quality score is beyond your 30. So I told you it should be beyond 30 or maybe equal to 30, right? That is why here it is green color. If you have a bad quality, how it look like, just let me tell you. Can you see here this bad Illumina data?
It goes to the red.
Exactly. Can you see for this data, the basis? So this is your one read here, and this yellow color, it is indicating what the each base, single base here. Okay. And you can see maximum bases are falling here in the red color. Can you see from here? So
Yeah.
that is why it is having what? Poor quality, because it is falling in the red color, right? That is why it is red cross here. Are you getting the difference between the good and the bad here?
Yes.
Yeah, in here I
Sakshi,
see
did you got this?
more than 50% has bad.
Yes, sir. Yes, sir. Maximum. Can you see that? Maximum they are falling. And even in the orange also. Can you see that?
Yeah.
Okay. Now, if you want to read about this like more in detail regarding this summary, I will tell you how to get that. So just type FastQC in the browser and you will get the copy of the FastQC. And go to the analysis module. So one by one, you can read it if you want to read. Okay, from here. Just for your reference, guys. Clear?
Okay. Okay.
So again in the per tile sequence quality also, here you can see entire graph is blue color. If it is blue, again the quality is good. But sometime like you are getting this spot here also with the different colors. So that means it will indicate your average quality also and the poor quality also. But if you need a good quality, this tile should be proper blue color. Okay, that is why it is giving you green tick. Then next is the per sequence quality score. So here we are going to check the quality according to your read sequence. So again on the x-axis, this is your Fred quality score. And on this y-axis, it is indicating how reads are distributed overall. Okay. So now can you tell me what is the Fred quality score now again for this reads?
So 36 only.
36.
37.
Yes. Yeah. So you can see in this graph, the peak, the maximum peak, it is taking at 36. Can you see here? The sharp peak here. So it is indicating what? The Fred quality score. It is what? 36 here. Okay. And also, you know, the Fred quality score cutoff should be more than 30. So the peak For overall all the reads, it is falling at the 36. So that is why it is having per sequence quality score is good here. Okay. So directly, like we can say from all the read, if the read sequence is good, that means the base is also having a good quality. Okay.
Okay.
The next, per base sequence content. So how are different bases? So you can see the different bases here. They are indicating with the different colors. Thymine with the red, C with the blue, A with the green, and black with the G here, right? So now you can see here, we are getting a random distribution. Can you see here? Random distribution here.
Yeah. To the
So
adapter.
what are those random distribution? Can you tell me any idea?
I think since it is at the beginning of the sequence, I will say that it is the adapt. It can be the adapter.
Okay. What about others? Any idea?
These are different bases.
Mm-hmm. Yeah, these are different bases. So actually this random distribution, why it is created? Because there are some biasness in your data. So because of the biasness in your data, it is creating this type of random distribution. Okay. So this is huge, okay, between the bases. So that is why here red cross. So normally if you want the good quality for the per base sequence content, it should have parallel lines like this. Okay. Did you got this graph?
Okay.
So where does the biasness come from in our data? Like, is there a reason for that?
Yeah. So we will discuss this biasness during the cuffling, sir. Okay, in detail.
Okay. Okay.
Yeah, maybe like in the wet lab, whenever people are doing the over amplification, the PCR step, right? So
Okay.
sometimes because of that also, like you can get those biasness. Okay, and there are or also there are a lot of other reason also that we will discuss during the cuffling, sir.
All right.
Okay. Yeah. Next, the GC content. Now, can you tell me why they have calculated the GC content? Any idea? Why not they have calculated the AT?
For stability, ma'am.
Yeah. So you already know this GC, it is connected with the 3 hydrogen bonds, right? And
Mm.
it gives you the stability for the DNA. Okay. So that is why they are checking the GC content. Now you can see the blue color. So this is like already calculated, okay, the GC content. But for our data, this is the red color line. Okay. So why exclamatory? If it is properly bell form shaped, then it will give you the green tick over there. But somewhere at the top, you can see there are short peaks also, right? It is not exactly like the bell form, right? So that is why it is giving you the exclamatory sign. Did you got it? So there is some differences, okay, between these two lines. Did you got this per base, like per sequence GC content?
Yes,
Yeah.
ma'am.
Any one of you? Did you don't understand? Just tell me.
Professor, if GC content is high, it means our DNA or RNA quality is much better. Yeah.
Exactly. Exactly. Now, what is N content? Any idea? Have you ever heard this N content?
Yes, ma'am. The machine was not able to read the base.
Exactly. So N content is what? Something gap in our data. So let's consider like during evolution, there is a small stretch of sequence got deleted. So that time it will create a gap. And so whenever we are doing or whenever we are going to do sequencing, that time sequencing machine is not able
Yeah.
to read your actual base. If the machine is not able to read your base, then it will going to create what? The N content. So in this graph, it is clearly seen like it is there is no N
Flat.
content, right? It is just falling at 0. So that means there is no n content in your file. Clear? So yeah, so rest of the part will continue tomorrow because I have a session. So we'll meet tomorrow. So meanwhile, what you can do, like you can perform till the fast QC step. Okay, everyone. And also try to download the raw data. Okay.
Okay, Professor. Professor, can I ask a small question?
Yes, sir. Sure, sure.
Now you receive the data from the online platform like NCBI. If we, for example, send our sample and we receive a sequencing or fast queue
Mhm,
from the company,
mhm.
then the method is like that. We put in the folder and then we.
Exactly. So
Okay.
let me share my screen. Like
Mhm,
whenever like you are doing the wet lab procedure, then you are
mhm.
sending to the sequencing platform, right?
Yeah,
Because
yeah.
if you do sequencing, it will cost you high. Okay.
Ex-
So
yeah.
after sequencing, what type of data you will get?
It's
This.
fast queue. Yeah, yeah.
Exactly. This file you are going to get, sir. FastQ
Ex
file. Okay.
exactly. Yeah.
Yeah. So from this, you are going to do the further analysis by yourself, sir.
Yeah. Thank you.
Did you got it? Okay.
Yeah, yeah. Thank you. Thank you.
Okay. Okay then. Bye bye. Yeah, we'll meet tomorrow, guys.
Thank you, ma'am.
Okay. Bye
Yeah.
bye.
See you tomorrow.
Yeah.
Thank you.